Thank you. So for the next 20 minutes or so, I'm going to talk about computational vaccine design, and this is joint work with Nora Toussaint and Pierre Dennis, who are also in the audience, so feel free to talk to them or to me about this later. So vaccines are one of the key achievements of medicine, one of the big success stories. But as you well know, there aren't vaccines for everything. Swine flu is just one of the latest things where you can see that there is a need for new vaccines, also for the quick development of vaccines for emerging infectious diseases, and also for personalized medicine. Personalized medicine means we also need personalized vaccines. And there is a need for computational vaccine design that might help to speed this process up of designing and developing a new vaccine. So when we talk about vaccines, what's really going on there? Basically, most of the vaccines that are on the market today are still based on the initial ideas of Louis Pasteur, who found that attenuated pathogens, basically heated pathogens, can be used to produce immunogenicity when vaccinated with this attenuated pathogen. So basically, the immune system learns to recognize the pathogen. It is not infected because it is attenuated. And afterwards, when exposed to the real pathogen, the immune system can react strongly and quickly and thus prevent the infection. If we look at this on the molecular level, then some of the key players are T cells, and the immunologists among you will hopefully forgive me for being very brief on this. It's a bit more complicated than I sketch it here. But in general, what's happening is that this recognition of self and foreign, which is done by the T cells, happens in a way that the antigens, which are part of the pathogen, for example, viral proteins, are processed inside a cell into smaller fragments, peptides, which are then presented on the cell surface by MHC class 1 and class 2 molecules. And the complex of MHC and peptide is then recognized by the T cell, which then basically produces the immune response we're after. So part of, if we look at this a bit closer, is that then we see that the antigens aren't really what causes the immune response, but it is just a part of this antigen, these peptides that are being presented. And not every part of the antigen is being presented. Here you see a protein from HIV, and highlighted in yellow here you see the part of HIV that is the peptide that's actually being presented in most of the patients on the surface, presented by these MHC molecules. So these T cell epitopes are usually short peptides, 8 to 20 amino acids on average, and they are presented by MHC, and this presentation is needed to cause a T cell activation and thus the immune response. And for predicting these epitopes, we have a whole range of methods that have been developed basically over the last 15 years, so we can predict these epitopes rather well, or at least what is being presented by the MHC. And these MHC are a bit problematic when it comes to vaccine design. MHC is a highly polymorphic gene locus, and that is good too because it's part of why the immune system is so successful. But when we design vaccines, this can be quite an issue. There are several thousands of different alleles of MHC class 1, class 2, and these alleles of MHC all present different peptides. Now, if you look at these allele frequencies, they differ widely by ethnic group or population. And these are the allele frequencies of 6 of these MHC class 1 alleles, A01, 02, 3, 11, 23, and 24. And this is what it looks like in Europe, and this is what it looks like in Southeast Asia. There are databases of these allele frequencies. And you see that if you design a vaccine that is based on epitopes being presented by A1 or A3, this vaccine will be effective in Europe because the allele frequency is rather high there. But if you use the same vaccine in Southeast Asia, you see that you will basically get 0 response. So that means if you design a vaccine, you will always have to have a certain population in mind, or if you do personalized vaccines, a certain patient even. So epitope-based vaccines are a slightly different idea, deviating from what Pasteur suggested. His idea was this attenuated pathogen. So basically, if we're talking about a virus, but similar things apply to anything else, also to cancer vaccines in the end. But this route here, weakened virus, which is then used as a traditional vaccine, also means that everything inside there is included, also possibly infectious particles, which is something you don't want to have in a vaccine. So what you can do instead is a more rational approach where you focus on the epitopes which actually convey the immunity. So you take the sequence of the virus, you sequence that, you get the sequence of the viral proteins that are the potential antigens. You can predict, using computation methods, the epitopes that can be responsible for the immune response. And then you need to select the epitopes and put them into an epitope-based vaccines. And I will not talk about how they can be constructed, but they could be as simple as just taking 20 peptides, synthesizing these peptides, putting them into a vial and using them as a vaccine. There are other ways to deliver that and lots of issues with that too. But basically what we will talk about here is the problem of selecting these epitopes, because you cannot put an arbitrary number of epitopes into these vaccines, but for example, to reduce production costs and also simplify testing and approval, you want to keep the number of epitopes pretty low. So this epitope selection problem is an optimization problem. If you talk to people in industry who are designing these epitope-based vaccines, then they tell you, well, we look at all the epitopes that we know, and then we sit around the table with a couple of experts for a couple of weeks, and we select these epitopes. And as a bioinformatician, you might argue, well, there must be a way to do this in an optimal fashion. Now, basically, the optimization problem is rather simple. We are given a set of antigens, and from these we can determine the candidate epitopes. We have additional information, for example, which epitope belongs to which antigen, the population coverage, also the conservation. Are some of these epitopes prone to mutation in viral genomes? That can be an issue. We also have a population which we want to optimize the vaccine for. So basically, we have for each MHC allele, we have the frequency of this allele in the population. And then we have a magic black, or in this case, rather white box, where all this goes in, and out comes an optimal set of peptides. And you, in the ideal case, in an ideal world, you would just synthesize these peptides and you had your vaccine. It's so very straightforward. Now, formally, we are given a set of antigens and their sequences. From this, we can compute the epitopes. We are given a set of alleles and their frequencies, the target population, and the goal is to identify a minimal set of epitopes yielding the best vaccine. And I kept this a bit fuzzy, intentionally fuzzy, because what is the best vaccine? And obviously, also, if you talk to immunologists, it is not exactly straightforward to define a good vaccine. And these definitions are highly controversial, and there are lots of issues with that. And that's one of the reasons why we decided we're not going to solve one problem, but we're going to present a mathematical framework that allows you to formulate different flavors of the problem, different requirements, immunological requirements to this vaccine. And there are certainly some properties that are of interest to everybody who wants to design a vaccine. Of course, you want to reach a good overall immunogenicity. That means that the vaccine is capable of inducing a potent immune response. You also want to have mutation tolerance if you construct a viral vaccine. Some of these epitopes can be destroyed by point mutations in viruses pretty quickly, and that would lead to immune escape against this vaccine. You also want to cover a huge population. And we know these allele distributions differ, so we need to target a precise population. And we also want to cover antigens. If we select epitopes that all come from the same antigen, we will have a less broader coverage of the whole pathogen. Again, that might be a problem. And there are various requirements that you can formulate and that you find in the literature about what makes a good vaccine. There's, interestingly, very little work on this. So far, probably the most important related work is by Vida Chalit et al. And they suggested a genetic algorithm that produces an optimal polypeptide. So this is basically a string of individual epitopes. They have a scoring function that aims at high antigen coverage, high allele-wise coverage, and they also consider only peptides that are highly conserved and likely to result from antigen processing. Having worked with combinatorial optimization for quite a long time, we followed a slightly different approach. Instead of using a genetic algorithm, we used an integer linear program, an ILP. So we formulated this epitope selection problem as an ILP, which aims at maximizing the overall immunogenicity for a given target population. The advantages of integer linear programming over genetic algorithms are, of course, that we obtain an optimal solution. So given this objective function, we can be sure that there is no better set of epitopes that would yield a higher immunogenicity. And also ILPs are very neat mathematical tools, so we have a convenient, flexible, and also very established mathematical framework. We can write these constraints down very efficiently. There are a couple of key assumptions we need to make, and they are also made by others. First of all, we assume that the epitope immunogenicities are additive. You might argue about that. There are some things going on that are not exactly additive, but this is the best model we have so far. We also assume that an allele's contribution to overall immunogenicity is proportional to its frequency in the population. So basically our total immunogenicity is a measure of the immunogenicity on the whole population, on a representative set of the population. And we assume that we can approximate the immunogenicity by the MHC binding strengths, but this is not necessary for the formalism. When we try to maximize the overall immunogenicity, we basically assume that each epitope E has a certain immunogenicity for a given MHC allele A. So we need to determine these IEAs, which are the immunogenicities of epitope E on allele A. And we simply multiply these immunogenicities with the frequency or probability of an allele A, and thus the overall immunogenicity is just the sum over all alleles over all epitopes of the immunogenicities and their probabilities. There are a couple of caveats. These immunogenicities can be experimentally determined, but in most cases we just don't have the data, and we don't have good predictors of immunogenicity yet. So what we do is we approximate this by MHC binding affinity in case we don't have the experimental data. Now, if we formulate this as an ILP, then we need to maximize the immunogenicity. So basically we take the definition of the total immunogenicity and we just add binary decision variables XE up here. And these decision variables become 1 if the epitope E is part of the optimal solution. It becomes 0 if they are not. And this is the job of the ILP solver to determine the optimal values of these XEs. And the solution of this ILP is then a set of epitopes to be included, and those are just the epitopes for which these XE become 1. Now, we need to formulate, besides this immunogenicity, we need to formulate these immunological requirements I mentioned earlier. And we formulate these as constraints that are added to this integer linear program. So we maximize the overall immunogenicity subject to some constraints. For example, we can say we don't want a vaccine that contains more than 10 epitopes. And this is a very simple constraint. We just sum over all epitopes and sum up these XE, and the constraint ensures that we get at most K XE that are set to 1, so we limit the number of epitopes in our solution. Slightly more complicated constraint ensures that at least tau MHC, a certain threshold tau, alleles are covered by the selected set of epitopes. And here we have to introduce additional variables YA. I'm not going to go into the details, but again, this is a very straightforward ILP thing that is often done. With this, we can ensure that we get sufficient coverage of the alleles. Similarly, we can ensure that each antigen is covered by at least 1 or at least tau A epitopes per antigen. Here we sum over all XE belonging to a certain antigen, and we add a constraint for each of these antigens, and thus ensure that each antigen would be covered by a certain amount of epitopes. And in a similar fashion, we can basically formulate all these immunological requirements in a very straightforward mathematical manner, very compact, very easy to read. And this is what we can put into this framework. Basically, we describe how our vaccine should look like from an immunological point of view. We add the required constraints, and then we solve this problem. We have implemented this using the C++ interface of Cplex, which is a commercial solver, but you can put in any ILP solver that is working reasonably well. We also tried it with a GNU linear programming toolkit for our web server. So this is pretty straightforward. The problem sizes that are of interest for vaccine design are rather, I wouldn't say small. It's not exactly a small problem, but they're manageable. We have hundreds of antigen sequences and usually dozens of alleles, and these problem instances can be solved with Cplex in a matter of a couple seconds, so a second or 2 or 3. That's usually all it takes to determine the optimal vaccine. Now we can compare what comes out of this with other approaches. So Wiederschalit in their paper, they had an example for hepatitis C virus vaccine design for that, which was based on 4 subtypes of HCV and 10 antigen sequences from the virus. And the immunogenicities of each epitope were determined using the biomass matrices. And as you would expect, if we look at the immunogenicity you achieve with a certain given number of epitopes, you see that as you add more epitopes, the overall immunogenicity goes up, which is not very surprising. This is overall immunogenicity, this is number of epitopes. And you can see that compared to the genetic algorithm, at the same number of epitopes, we achieve a much higher overall immunogenicity. Caveat here is that their objective function was also slightly different, but even using their objective function, we can achieve better results. Or if you look at it from another point of view, if you want to minimize the number of epitopes, then you can achieve the same immunogenicity they achieve with 24 epitopes with 18 epitopes. Yeah. So these are the two sorts of ways you can compare these results. You can also compare it to other strategies for selection of epitopes. So again, we try to estimate the immunity or maximize the immunogenicity given a set that we need a set of at most 10 epitopes. And you can either use the optimal strategy, this ILP strategy, which is here labeled with BOI, best overall immunogenicity, and we plot here the overall immunogenicity of this set of 10. And you can compare that to a greedy approach where you just take the best epitopes out of your set. And that gives about half the immunogenicity with the same number of epitopes. Or RSE, this is a random set of epitopes, so you take all the good epitopes, only the best epitopes, and you randomly select from these epitopes. And that gives you, again, lesser overall immunogenicity. Or you can turn it around again. You try to determine the number of epitopes that you need to achieve a given overall immunogenicity. And again, you see with 5 epitopes, we achieve the same immunogenicity that you can achieve with taking the 12 best epitopes. So this works reasonably well. We have implemented this also as a web server, which is called Optic Hope. And here you just paste in the antigen sequences that you're interested in. You then can select the target population, say you want to optimize your vaccine for North African population or any other. We basically take the allele frequencies from a database, and or you can enter the allele frequencies manually. Basically, these are the two parts of the input. You define the antigens, you define the allele frequencies, and then you need to define a couple of constraints. For example, you require that you have a minimum conservation of the epitopes, that you have at most 10 epitopes in your vaccine, that you cover a certain number of alleles, a certain number of antigens. You can simply add these constraints, turn them off if you don't like them. And then you click on next, and what you get is another page yielding the results, basically the vaccine. So you get a list of the epitopes, 10 epitopes that should be included in the vaccine. You see how much each vaccine contributes to the overall immunogenicity, like this here, 26%. You see which alleles are covered. You see which antigens are covered. and you get some statistics. So we covered 19 out of 19 alleles, 5 out of 10 antigens, and we covered 99.63% of the population with these 10 epitopes. Now, if we look at this whole setup, then one of the obvious things that need to be improved if you want to go to a real vaccine design is that we need to get better numbers for these immunogenicities as opposed to MHC binding. I said we can get these numbers experimentally, but that is a lot of work. It's not easy to get these numbers. We also are working right now with experimental groups on getting quantitative data on immunogenicity that would confirm or give us an idea of how valid this hypothesis of the additivity of these individual immunogenicities is. That is not really straightforward. We see that pools of peptides behave slightly different than individual peptides, which is not exactly surprising, but so far we don't have a better model than this additivity, so this is what we stick to. And hopefully also animal and clinical testing of the epitope-based vaccines that are now in preclinical and clinical phases of the trials will yield data that allows us to identify good scoring functions or better constraints for designing these vaccines. And also an interesting application will be cancer vaccines and personalized cancer vaccines where this might be a very promising strategy to select the epitopes for a specific patient. With that, I'm at the end, and I want to thank the people who did most of the work, Nora Toussaint and Pierre Dönnes. The whole project was funded by the Deutsche Forschungsgemeinschaft, DFG, as a Collaborative Research Center 685 Immunotherapy. Thank you very much.
Hi, very nice talk. Just one or a few questions, actually. So in order for a peptide to be presented, it's not enough for it to bind MHC. So even if you have a set of optimal peptides, you still need them to be cut eventually.
Yeah.
So can you make an optimization procedure?
I mean, you can predict, basically the two steps of class 1 antigen processing are TAP transport and proteasomal cleavage. And there are computational models for that. Actually, it's not on the slides, but we have that in the paper, and you can look it up there. It is, you can estimate that, but the caveat is that the predictors for that are not as good as we would like them to be. But you're right, antigen processing is, of course, a step that should be and can be included.
But the question is slightly different. So within the optimization function, assuming you believe the algorithm, can you simultaneously optimize for immunogenicity and the fact that your set of peptides will be positioned in the way that they'll be cleaved at the end to really produce epitopes?
Well, are you saying, are you interested in how the epitopes should be put into the final vaccines, or?
Precisely.
Yeah, I mean, that's a different problem. Yeah, here we are only selecting these epitopes. I think what you're referring to is how do you string these epitopes together in a way that they are then produced. So basically, do I do a peptide mix vaccine, which is one way to do it, or do I design also the spacers in a polypeptide vaccine? And that's a different optimization problem, but that can be solved actually in a very similar fashion too.
Okay.
I wonder when you do class 1 vaccinations, whether you also get humoral responses. When you immunize with a vaccine which is binding to MHC class 1, whether you also expect to get antibody responses under this type of vaccination, or do you have?
It is usually observed, yeah, but obviously it is not necessarily you get a humoral response for each of the epitopes. But yeah, you observe humoral responses too.
And in case of cancer, would you go just for class 1 or class 2 as well, or a combination of both?
From literature, we know that the combination of the two is best. You need CD4 and CD8 responses. And I know at least of one clinical vaccine that contains both class 1 and class 2 epitopes. But it is unclear at this point how you should balance the two. I mean, how valuable is one class 2 epitopes compared to one class 1 epitope. And these are all things that we don't know, and we need to analyze the outcomes of these clinical studies very thoroughly to actually get an idea of how to include that.
Last question, how do you get to the targets in cancer therapy?
Well, in the clinical studies, we are at least involved in that collaborative research center. This is based on the analysis of the ligandome and the analysis of the transcriptome that is gotten from the patient.
Maybe I ask a very last question. What about side effects? Are you afraid of side effects?
I'm not afraid of side effects. I leave that to immunologists to have a look at that. We are just interested in selecting the epitopes at this point. No, I mean, obviously you could have side effects, but part of this epitope-based vaccine strategy is to minimize side effects by just including the epitopes and not have dirty vaccines that contain basically everything that was in a tumor or everything that could be in a virus. But yeah, we just don't know about possible side effects at this point.
Thank you very much.